The capsules we call crapsules. That's frozen poop that can make it through the onslaught of enzymes and acids that are in your stomach and in your small intestine. If they make it to the colon, they've made it to the promised land. And that's what you're trying to do.
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. My name is Jesse Lawler. I am your host, and this is the 162nd episode of this podcast dedicated to the ongoing betterment of your brain by any and all means at your disposal, even and especially using asymmetric, unexpected, and counterintuitive means of getting the brain to operate better, like paying close attention to your digestive tract. We are probably a little late to the party here, given that we're 162 episodes in, and we haven't really talked about the major connection between brain health and gut health, but we're about to solve that problem. We're going to be talking about guts, the microbiome, and some of the amazingly bizarre statistics and correlations between what's going on in our stomachs and our brains. I'm using stomach in the little kid sense to mean basically everything below your lungs. We're going to be talking with science author and researcher Scott Anderson in the main interview, getting into topics as far astray as serotonin and consumable fecal pellets, so brace yourself. If you hang around until the very end of the episode, in the Ruthless Listener Retention gimmick, I'm going to give you a reason to feel a certain amount of kinship with the schizophrenics out there. This assumes that you're not schizophrenic yourself, which statistically speaking, some of you probably are. But sometimes some of the symptoms of what is generally a very unfortunate condition like schizophrenia can manifest fairly benignly in everybody else, and researchers have stumbled onto an interesting one, an example you'll probably find close to home if you hang around for the Ruthless Listener Retention gimmick. But as usual, let's kick things off first with This Week in Neuroscience.
Smart Drug Smarts, This Week in Neuroscience.
So relatively recently, back in episode number 156, I spoke with Dr. David Bellinger about lead poisoning, lead toxicity, and all the nasty stuff that the element lead does for the human brain and nervous system. It was in the news a lot last year because of the Flint, Michigan environmental fiasco. That is far from the only fiasco involving lead, but overall, lead has got to be said to be a fairly major public health success story, just given that even how terrible it still is, it's a lot less terrible than it used to be. Airborne lead particles, and of course there are particles of lead that are not airborne, but airborne lead particles fell 94% between 1980 and 2013, thanks to lead having been phased out of automobile gasoline within the U.S. And there's every reason to suspect that it's had dramatically positive results both as far as people's brains and the downstream effects of people having better brains. There are even those who speculate that the drop in violent crime during the 1990s is associated with the declining use of lead in the previous 20 years, and that the number of potential violent bad guys hitting the street was reduced because some of those bad guys were acting out because they didn't have quite as functional of brains as they should have had if they weren't in a high lead exposure environment. But with all these positive things I'm saying, I'm sure you're waiting for the but. And it is this. Stop for a second and think where the major source of airborne lead is now, 2017. It might not be what you guess. It's not cars anymore. It's not home fix-it people sandpapering lead off paint chips and old walls. It is actually something called avgas, aviation gasoline, which is not the same gasoline as people put in their cars. And in fact, it's not even the same gasoline that commercial airlines use for their fuel. Commercial airlines generally use a higher grade of gasoline that has had lead taken out of it over the years, but there are apparently some 167,000, give or take, small private planes in the U.S. that are using the only gasoline that is currently available for them, and which still does contain lead, and is now the greatest environmental source of new lead entering the atmosphere. A study conducted recently at MIT has looked at the amount of lead, which is still quite significant, which is getting dumped into the air. As of 2008, there were 248 million gallons of this avgas that was used. Not all of which is lead, of course. Most of gas is made up of hydrocarbons. But nevertheless, they ran the numbers looking at the amount of airborne lead that is getting disseminated out there. Using a couple of different statistical models, they figured that looking at the effects of people's IQ from lead exposure and what that translates to into economic productivity, the American economy is losing about $1.63 billion annually because of unnecessary lead exposure. And American workers themselves are losing over $1 billion in lost earnings. And that was sort of the bottom of the lead barrel as far as estimates. Some of the researchers' models put this number as high as $11 billion. So the regulatory game of whack-a-mole reducing lead in the environment is a continuing saga, but things are going in the right direction. It's unlikely there's going to be any new additions of major lead sources that would get approved by any government at this point. But funny, weird, and kind of scary to think that recreational flying vehicles are essentially slowly, minorly, but significantly poisoning us little by little.
Smart Drug Smarts, the podcast so smart, we have smart in our title twice.
Picked up a couple of 5-star reviews on iTunes. Listener Zal83 from Australia said, randomly came across this a few weeks ago, and it is Joe, one of my favorite podcasts. I don't know if that's an Australian term, Joe. It's written like the guy's name with a capital J right in the middle of a sentence, but maybe we've just picked up some Australianese. And listener Smeinzer from the USA says, conversations with scientists and doctors in a way that makes it easy to listen to for someone without a medical or science background. Well, thank you both very much. Thanks to everybody that's been leaving iTunes reviews or otherwise disseminating Smart Drug Smarts out there to the public. This is the very first episode of the new year. Happy New Year, everybody. And later today, I'm going to be pressing publish on the first Brain Breakfast newsletter of the new year. You've probably heard me mention it before, but if you haven't signed up yet, I invite you to do so over at smartdrugsmarts.com/newsletter. It's one of the other appendages of Smart Drug Smarts besides the podcast itself. Another appendage are the supplement stacks over at Axon Labs, Nexus, and Mitogen. If you're entering the new year bleary-headed and not quite all you could be, still haven't fully recovered from New Year's Eve, anecdotally, this is not a tested statement. I've been told that Mitogen is actually pretty good in helping people bounce back from hangovers. It is a general mitochondrial health supplement, has ingredients that will both encourage the production of new mitochondria and also give your existing mitochondria the biological precursors that they need to make ATP. How or why extra ATP might be a hangover solve, I leave it for somebody else to figure out. There actually are some really interesting chemical means of hangover avoidance that science is poking around with. I feel a little bit mixed feelings about that, simply because avoiding the hangover doesn't necessarily mean that you're avoiding the neuronal damage of alcohol itself. But even saying that, I feel like I'm being a moralistic old fuddy-duddy, so consider the jury still out here. But anyway, Nexus and Mitogen are both available over at axonlabs.io on the web, or if you happen to be loitering around the smartdrugsmarts.com page, we have a shop link in the menu that'll get you over there also. That's all I've got in the way of mini announcements, so we'll get right into the interview. Lots of good stuff planned for 2017, so stay tuned.
Smart Drug Smarts.
Okay, so I'm about to be speaking with Scott Anderson, who is an upcoming author of a book called Psychobiotics. I'm pretty sure that's still going to be the title. The book's not out now, but when it publishes later this year, we'll go back to this episode's page and add the link there. So if you're a latecomer to this episode, you can find the link now. If you're listening on the day of publication, you, like I, will just have to wait. But this conversation will be a great sneak preview of a lot of Scott Anderson's work and research, how his minding his own business dealing with horse ulcers led him into the deep, dank, and surprisingly interesting reaches of the human digestive tract and right back on up to our brains. You'll probably get more fart jokes in this particular edition of Smart Drug Smarts than you will in probably anything we've published to date, but it's hard to avoid these things when you're talking about digestion, so cut us a little slack. I also realized, just re-listen to this interview, there's one terminology bomb that we drop, that is C. diff, which stands for Clostridium difficile, which is a bacteria that causes infectious diarrhea, not something you want to have. When you hear C. diff in the coming conversation, you can just think bad, which by default makes some of the poop-based therapies that we're going to be talking about the good guy in that particular battle. But with no further preamble, let's jump in with Scott Anderson.
I'm a science writer. I'm also a scientist, but not in a very relevant category. So I work with horses, and I've been working with horse supplements, and it turns out that horses have a lot of ulcers. And when I looked into it, I realized that a lot of this seemed to be caused by bacteria. So I started getting into the concept of bacteria being a major player in colonic upsets and other kinds of issues, and came across a couple of guys that were at University College Cork in Ireland. John Cryan, who is a neurologist, and Ted Dinan, who is a psychiatrist, started working on a project that they called Psychobiotics, and it totally caught my eye, and I started getting into it, blogging about it and writing about it, and I got in touch with them, and they had seen some of my blogs, and they liked me anyway, and so we got together and said, let's write a book. And the idea behind Psychobiotics is that these are probiotics that improve your mood, bacteria that make you feel better, and in a heads-up test with probiotics or these psychobiotics against antidepressants, the psychobiotics actually came out ahead. A lot of people understand now that antidepressants are, at least to some extent, a placebo effect. Some people are really benefiting from them. Others are finding that they're not very good, but they are excellent for putting weight on. And that's one of the issues that is actually germane to this, because the same bacteria that are in your gut are producing neurotransmitters and things like serotonin, unbelievably. 95% of the serotonin in your body is not in your brain, but it's actually in your gut. The gut is called the second brain, and there's a reason for that. It has as many nerves as your spinal cord does, and it's acting like a brain. Basically, its job is just to digest food, but that's still a reasonably good job for a brain to do. But the bacteria that are in there are doing a big part of the work too. And it turns out that if you have an infection or if you have an inflammation, a part of what the body does is to kind of shut down and and go into what you call sickness behavior. And sickness behavior, if you look at it, is, well, you can imagine it. It's like, all right, I've got a flu. I'm going to bed. I'm going to cover myself up. Leave me alone, which in a way looks a lot like depression.
That is an interesting point.
And so a lot of people have started to put this together, the idea that you've got bacteria that could be causing an inflammation and the idea that you have depression or anxiety is actually sort of reasonable. Maybe from an evolutionary point of view, it's a way of saying, look, I'm sick, I'm contagious, get away from me and I'll get away from you. And from an evolutionary point of view, it might actually make some sense to say, look, you're sick, get depressed. And for people who have ongoing chronic inflammation, it could be that that's a big part of the cause, the source of their depression. So we're looking into that. It's kind of interesting. Depression is comorbid with many diseases of the gut, including irritable bowel disease and Crohn's disease and many others. And it's possible that if you just fix your gut with the right psychobiotics, that you could actually cure your depression and or anxiety.
Do they have a fairly good handle on how to diagnose what gut bacteria a given person needs and how they might approach that?
The problem is basically solved in mice. So if you have a depressed mouse, we can fix it. The studies with humans are ongoing, and we're looking at what's coming out now. Directionally, it's looking good, but it's hard to give anybody a real good recommendation on a specific probiotic for depression until these studies are a little bit better. So I'm holding off on that. But I can see the big picture here is that things like Lactobacillus rhamnosus in a mouse can actually cure what a mouse version of depression. And there's an issue, of course, in trying to see what a mouse version of depression is and how that really relates to people. The same thing with autism. There is an autistic mouse model. They can give specific bacteria to this autistic mouse, and they can roll back a number of the issues involved with autism. It's not a complete cure, but it's an amazing improvement, at least in these mice. And so translating this over to humans is going to take a little bit of time, but the evidence so far is quite compelling. And with humans, we are seeing some slight improvements with some early studies. So the first human study was done with yogurt, and they took people who were not depressed and gave them yogurt, and they measured their levels of happiness. And they found out that those who were on the yogurt were significantly happier. Really, the study showed that there was a slight increase in happiness. So it wasn't like a gigantic leap. But something does happen, and we know that those particular bacteria are not what we would expect these specific psychobiotics to be, but they are related. The thing about stuff like yogurt and other things that you might eat, fermented foods and stuff, is that they are, by and large, transient creatures in your gut. So they're just passing through, and they are not necessarily going to take up residence. So it's not clear that any of them are going to stick around, and that's an issue. To some people, that's a good issue. It means that if we do find the right psychobiotics, that you'll be hooked on them forever, and somebody will make some money. But what would be nicer would be a way to make your own guts work better to begin with. And for that, we're looking at more like prebiotics. And those are the substances, the foods that feed the bacteria. And with the right prebiotics, you can increase the right probiotics in your gut and hopefully raise those levels to a point that you can get psychobiotic effects. And it's something that I feel comfortable with there, is the idea of recommending not necessarily pills, but foods that are rich in certain specific prebiotics, including something called fructooligosaccharides and galactooligosaccharides. These are complex sugars, and when you feed them to your colon, basically, it enhances the populations of bifidobacter and other beneficial bacteria. And so that's one possibility. Now, it's not clear yet, again, though, that anything is going to really be permanent in that regard. And this is where perhaps the most far-reaching therapy is, and that gets back to the poop that we started with, is the fecal transplant. And again, the data here are a little bit iffy. We're still looking at how all of this is going to play out. But we have some interesting reports of people who were depressed. They got a fecal transplant, and within minutes, they were no longer depressed.
Within minutes?
In minutes. It seems like after about an hour or so, most of these people were like, I'm a new person.
Can you describe the route by which serotonin, for example, produced in the gut actually makes its way to the brain? Because if we take these people at their word and say that within minutes they're actually having a mood change based on a fecal transplant, I'd love to understand what the mechanism that could cause that would be.
Well, that's a really good question. I don't think it's happening that way. It's true that these neurochemicals, neurotransmitters, are being produced by bacteria in the gut. There is GABA, there's serotonin, dopamine. All of these things are actually being produced by bacteria, which seems kind of shocking, but it turns out that these are not necessarily very complicated chemicals, and they may be using them to talk to each other. None of those things should get through the blood-brain barrier. So it's not clear that that's what the actual mechanism is. The mechanism is probably more related to cytokines and the immune response that your body has. And that can kick in very quickly. The levels of cytokines in your body can change within minutes of reducing the cause of an inflammation. So if you get rid of certain pathogens, which a fecal transplant does instantly, if you get rid of these pathogens and substitute that with a good system, your immune system can reset very quickly. And then cytokines stop circulating through your blood. And that may be one of the mechanisms that's important. We know that the immune system is important. We know that the vagus nerve is also important. And the vagus nerve is a direct connection between the hypothalamus, which is in your brain and controls things like hunger and fear and things. And it connects up to your stomach through the vagus nerve. And that's another possible connection. We know that if you cut the vagus nerve, that some of the effects that we're seeing go away. And some people actually are trying to cure intractable depression by cutting their vagus nerve. With some of them it works, but it's surgery. It's certainly not the first thing you'd want to think about. But these are people who have been fighting for their life with intractable depression. They are desperate. And there are a lot of people who are affected by depression. It's surprising how many people there are. The WHO says that there are 350 million people with depression, and they say it's the number one cause of disability in the world, which is a little bit shocking, and you kind of wonder how that has happened and what's going on with all of us to put us in this state. And at least a flip answer would be that the Western diet is not supporting the kind of bacteria that we need basically to keep our inflammation levels low. So we're inflaming ourselves, and we're inflaming ourselves potentially with refined starches. It's not clear how much of that's true, but it's certainly clear that since we've been taking more and more fiber out of our foods, that we've had more and more issues. That's correlational, though, not causational, and it's really difficult to get to the bottom of that because dietary studies are notoriously difficult.
Yeah, and we eat such a varied diet. It's a lot easier when you can feed rats rat pellets and that's all they get, but any given person eats food from all over the world. It's like I can be in Brazil eating Chinese food, and how do you test that?
Well, in fact, your gut bacteria can change themselves really quickly. Overnight, they can change themselves. And this, again, comes back to this idea that things can happen quickly. Your gut bacteria can change overnight.
Let me ask you a question about that, actually.
Yeah.
It brings me back to something I wanted to jump in on earlier. You mentioned most of the bacteria, I guess, living in our gut at any one moment are sort of on the choo-choo train out. They're not clinging to the sides of the walls and long-term passengers within our body. If that's the case, why is there the big concern about people using antibiotics and temporarily killing off their gut bacteria if all of these things are getting replenished by the food we're eating? Why is it a big deal if we kill what's in us at the given moment?
Well, the bacteria in your gut actually gets set up by about the time you're age 2, by the time you're weaned. And that bacteria gets set up and it finds its way deep into crevices in your gut, and it finds its way into places called crypts, which are pits that line your intestines. And they can live there, and then they can live in the mucus, and they can stay there, and they can repopulate yourself when you lose them. Some antibiotics, though, are really, really good at scouring them out. And the issue that we have with antibiotic treatments, a lot of times people are going to take antibiotics. They'll come back in a couple of weeks. Especially these days, they're encouraging people to take yogurt and to try and repopulate your gut with some sort of probiotics or prebiotics. They're trying to restore it. But the evidence is that every time you take antibiotics, you lose a few extra species. And the odds of you getting them back are really low because many of these species can only live in your gut. You're not going to find them in food. You're not going to find them through the air or in the dirt. They are very well tuned after about 12 billion years of evolution to live only in your gut.
So how are they finding their way into the guts of the 2-year-olds?
That's the amazing part of this, is that there's several places where these are happening, where the inoculation is happening. One of them is we are now finding that there's bacteria in places that we never thought that they existed before. One of them is in milk. Milk is perhaps the most important route for getting some of these bacteria into your gut. Your mother's milk is full of oligosaccharides, specific oligosaccharides, a lot of them, but they are not digestible by the baby, which is absolutely astonishing. The only thing that those sugars are for is your bacteria. The baby cannot handle those sugars, bacteria. Eat those sugars and then produce butyrate and propionate and acetate, other fatty acids that in turn nourish the baby. So that pure white liquid that you're thinking of, actually what happens is there are dendritic cells that go down into your mother's gut and they pull out specific bacteria that they want and bring it up through the lymph glands into the milk ducts.
It's almost like a spark plug for the baby's digestive metabolism.
Yeah, or or a starter kit, like a sourdough starter, and here it is being squirted out of a nipple, and it is astonishing. So if you weren't breastfed, you will have issues with your gut. Now, can you get it other places? Yeah, there's lots of places that you can get it. People are going to be kissing you. There are some ways that it can get transferred. Otherwise, you can take probiotics and prebiotics. And by the way, a lot of these things, these bacteria that get established are not static. So the population that you get when you're a baby is specifically to digest those oligosaccharides that's in the milk. As you start to get weaned, the bacteria in your gut changes. And in fact, it seems to precede the change. It seems to know that you're about ready to go to hard food now. And so it starts to change to bacteria that can digest vegetables rather than the ones that digest milk sugars. So you may lose some of those earlier ones. And that's why if you can make it through all of that, even if you were bottle fed, you can still come back and pick up some other bacteria later on. So it's not like you're doomed, but if you are breastfed, you are way ahead of the game. You've established a microbiome that's much more potent and much more resistant to depression and anxiety and further mental issues that come from inflammation.
And yeah, I mean, probably until 100 years ago, that was everybody. I'm not sure when infant formula was even created as a mass market thing, but I mean, it's within the last couple of generations.
Actually, it goes back to cow's milk and animal milk has been used for babies in the past.
Okay.
If the mother, for some reason or another, can't lactate, if she can't breastfeed, then you will try and find another human that can help. But if that doesn't work, then you can go to these animal milks. In around 1900, there was a French pediatrician named Tissier, and he looked at the milk and he saw they were having problems. The babies that were breastfed were doing fine. The bottle-fed babies were dying at a rate of 7 times higher.
Wow.
So he looked at what was in the milk and he found bifidobacter. And bifidobacteria is the one that is very popular in terms of digesting these oligosaccharides. And he added bifidobacter to cow's milk and it improved the health of these babies. It didn't cure everybody, but it did help. And that kind of was the beginning of formula. It was a little bit more like yogurt, but it was still the beginning. And people were starting to realize that some of these bacteria were important. Now, Tissier was wrong about certain things. He was right about the bifidobacter, and that seemed to be helpful. But he assumed that babies, when they came down through the birth canal, picked up a little bit of bacteria in the vagina, but that was it. And now we know that there's probably bacteria in the uterus as well. And in fact, we found bacteria in the placenta and in the amniotic fluid and in the baby's gut. So we're finding these bacteria where they shouldn't be. And it's because we have better ways of detecting bacteria.
Right.
But one of the caveats here is that the way that we find bacteria today is using PCR. And PCR amplifies the DNA. So you can find the DNA, you can say, yeah, there's bacteria there. But you really can't say that. What you can say is there's DNA from bacteria there.
That's a pretty strong smoking gun, though, if there were at one point bacteria in there.
Well, sure. I mean, if they're alive or dead, it still means that it's not sterile.
Yeah.
So it actually changed a lot of people's minds. Researchers are still reeling from this one. The fact that there's bacteria in the placenta is a little bit mind-boggling. And it doesn't seem to be always strictly associated with the pathogen. So we have known that bacteria can get in the vagina and into the uterus, but it's usually bad. So to see these commensal bacteria, to see the good bacteria, beneficial bacteria in the placenta was a shock. And we're still trying to digest all this information. By the way, a lot of this stuff is happening like this year. So you're looking at research that is so new that people have really not been able to incorporate it into their mindset and to, reset their expectations for what's going on with psychobiotics and probiotics and all of this stuff. This is an amazing new age where everywhere we look, we're starting to see bacteria.
Is there sort of like a zero-sum game, like only a fixed amount of space to live, like bacterial real estate within your digestive tract, like where if you're adding more of a certain type of bacteria that you're automatically squeezing out something else? Or is there plenty of room to go around?
There's plenty of room and they're small. They outnumber us, but you do have the right approach here because what happens is not so much that you're pushing it out space-wise, you're pushing it out living space-wise. In other words, certain bacteria will eat the food that other pathogens want.
Right.
And they will starve the pathogens out. And that's how things really work in the gut is it's a competition and you're just trying to encourage the good guys to win. But the good guys and the bad guys are often exactly the same bacteria. It's hard to just say E. coli is bad because E. coli is in everybody. C. diff. We know C. diff is supposed to be bad. We're all threatened. We're all scared to death of it. But it's a normal commensal in your body. It's only when things get what they call dysbiosis, where the relative profile of your bacteria changes, changes that some of these guys that we think of as pathogens become overrepresented, and that's when they become pathogenic. Normally they're fine, they play along well with everybody, but when they start to lose some of their competitors, they can take over and cause some real damage.
Right. It's almost like an algal bloom within your body.
It is. It's like a neighborhood where you just, this neighborhood is really set up and you could go in there and try and take it over, but it's going to be difficult because these guys have been in here for a long time. So that's one of the reasons why, for instance, the yogurts and things like that are not all that useful sometimes, is that they can't really get any purchase in there. So they can help you going through, and what they do when they go through is they produce these fatty acids that are beneficial. And by the way, the fatty acid of butyrate does get straight into your brain. It happens very quickly. That's probably one of the earliest ones that gets up to your brain and starts to make you feel better. Butyrate gets into your brain and actually changes the epigenetics of your brain DNA. So it starts to have your brain produce things like GABA. So it's an indirect way of it happening.
Yeah.
But you can see that there are these methods, in particular with fatty acids, that you can get straight to the brain and start to make some changes there.
Tell me about what a fecal transplant actually is. And I give you permission to go beyond the realms of G-rated propriety. We try to keep this a family show, but this one is probably not going to be possible to answer. I've heard about both capsules that are consumed and also others that are going in the outdoor, as it were.
Well, yeah, the capsules we call crapsules. That's frozen poop that we believe can make it through the onslaught of enzymes and acids that are in your stomach and in your small intestine. If they make it to the colon, they've made it to the promised land. And that's what you're trying to do. So the actual way that you do a fecal transplant is you start out by getting rid of your own bacteria. You take a very heavy dose of antibiotics. They clean out your gut entirely with the antibiotics. And then usually the way that it's done in the hospital is they use an enema. It's very important to get good poop. You can't just go to your neighbor and say, hey, can I borrow some of your poop? You don't know whether your neighbor is, for instance, he might be depressed, in which case you could wake up after this procedure. Actually, you're usually awake during it. You could come out of this procedure with depression. You could come out, one person got poop from an obese person and became obese. So a lot of the things that we think are really not relevant to your poop are probably relevant to your poop, and you really need to check it out. So there are places that are farming and banking poop, and they run it through a lot of tests to make sure there's no viruses in it, no bacteria in it. On some of them now, they're starting to do psychological profiles to make sure that you're not getting something from somebody who's really depressed.
To me, that just sounds like a just so story, like black cat walked across my path and then my mom died and it was the black cat's fault. One person getting obese after having a fecal transplant from an obese person, like, is that something which is happening consistently enough where the science is agreed upon? That's what's really going on. That just strains credibility for me.
I would agree with you on this one. The anecdotes are out there. We look at them and then we say, great, now can we do a study? And we're still at the very early stages on this. We're starting to buttress these anecdotal studies with some real data, but it's not all in yet. And I've talked to some of the people doing these studies at McMaster University in Canada, and they say, look, we're seeing stuff all the time. And it does seem to be that you can pick up things like depression and that obesity may be involved too. So I think that these things are kind of like a just so story in a lot of ways. And we have to be very careful not to oversell this stuff. I think that the microbiome has been overhyped considerably. And the problem with that is that it's probably going to end up being true. But in the meantime, if we start saying things that are not really well known, I think that we destroy our credibility.
Right. Yeah. Like eat goji berries and you'll live to 150.
Yeah. I'm not going to tell you that. I will tell you that these things are interesting and that they make you wonder about stuff. But what they really want you to do is to do an experiment. Yeah. So we don't have that yet. But we do have randomized controlled experiments in terms of defeating C. diff. Fecal transplants can get rid of C. diff. It is a 95% effective treatment. There is almost no treatment on the planet that is that good. And that is amazing. And it seems to last for up to like 6 years so far. They just keep looking at these people and they keep having the same microbiome. So it seems to be a successful transplant. And I consider this to be like an organ transplant. Your bacteria in your gut, it's like an organ. It's an unusual organ because it's also dissociated and a bunch of independent creatures running around, but it's still an organ and it's still yours. And you got it when you were about 2 years old and you've still got it unless you've had a lot of antibiotics. And so to wholesale take it out and then replace it with something else is kind of like an organ transplant. And I think that we want to be careful about that. That's not something you want to do idly. But if you're dying from C. diff, it is number one, the best, far and away, the best treatment you can get.
Yeah. Do we have a sense sense of how much a person's microbiome varies from one person to the next?
It's so variable, it's ridiculous. You can look at my bacteria today and my bacteria tomorrow will be different. Your bacteria today and your bacteria tomorrow will be different. Our bacteria will also be very different from each other. About a year or 2 years ago, they started working on something that they called enterotypes. They found 3 basic enterotypes. It sounded great. It was just great. It was the beginning of a new era. We could say, I'm A, I'm B, I'm C, I'm enterotype, whatever. And it all fell apart. All of the studies that came after that said, no, it's just not that easy. The amount of change between you and me and between me today and me tomorrow is so great that we really have yet to find the commonality that would let you even say that this is you from one day to another. And probably a large part of that is because a lot of these studies are done with fecal bacteria. And fecal bacteria are not really representative of the bacteria that are living in your gut. And to do that is more invasive. You got to go in there and scrape the sides, scrape the walls, and look at what's in there. And that's just not being done anywhere on a casual basis. And it's only rarely done when you go in for things like irritable bowel syndrome.
So what you're saying, the type of bacteria that are living in the lower intestine versus sort of higher up in the digestive tract, those are different landscapes from a bacteria's perspective.
Absolutely. You can see it change as you go through the gut. You can see that bacteria that live in your stomach, which is highly acidic. It's like battery acid in some ways, and it's graded, but at some point it's acidic as battery acid. Why? Why is that? Well, partly it's to digest your food, but almost certainly it's to kill bacteria. So a lot of bacteria get killed right there, but there are bacteria that can live in the gut and that can live in the stomach, and Helicobacter is one of them. And we now know that Helicobacter is probably responsible for most gastric ulcers. Cholera is kind of interesting too, because cholera bacteria are not bad. What makes them bad is a virus infection that switches on a gene that turns them into a pathogen.
Wow.
So the story is never as simple as you think it is. And by the way, almost all the studies that are being done right now are being done with bacteria. Very few people are looking at viruses. Very few people are looking at fungus. Those things are going to have a huge impact as well, but we don't know what it is. But it might explain a lot of the problems that we're having just looking at bacteria. If you're looking at bacteria and ignoring what are called phages, those are viruses that attack bacteria, then you're really missing a big part of the story. And so that's something that we need to go back and look at too, and we're just not there yet. It's a complicated story. It gets more complicated all the time. The more we look into it, the more we find. But as you go through the gut, you will find a different population of bacteria, each one finding its perfect range of pH and enzymes and things as they go down until you finally get to the colon, which is the promised land for bacteria. It's nice and warm, perfect place to do fermentation, and they love it there. That's where you're going to find most of the bacteria in your gut.
That's really interesting to me that a person's microbiome varies so much day to day, and yet there's also some element of commonality over the course of a lifetime that stays with you from the time you're 2 years old onward.
Yeah, and I think that the story there is that these bacteria can find a way to hide out. The fecal matter that's going through, a lot of it is tricky to nail down. Some of it's coming from your food. Your food has bacteria in it that you are not aware of. You are washing your vegetables, and that's doing nothing because bacteria live inside the vegetables. Bacteria live in the leaves of cabbage. They live in the leaves of spinach. They are not just on the outside. So you're getting a lot of bacteria at every bite you eat. Then you eat sauerkraut or you drink kombucha. You're getting more bacteria that way. And yes, probably 70% of them are being killed in the stomach, but that leaves 30% to get through and to try and get down to the promised land of the colon. When they get down there, they also have to put up with the ones that you've got established down there. So there's a big kind of a battle going on, and there are going to be casualties in there. So what you're pooping out is a lot of bacteria, a lot of dead bacteria that didn't make the grade, that got killed in the colon by other bacteria that said, hey, that's mine. So it's tricky to know by looking at fecal bacteria if we're really getting a good sense for the bacterial changes from day to day. I'm assuming that there is some pretty good commonality from day to day deep in your crypts and probably in your appendix.
That's interesting. In political history, they say that history is written by the winners, but maybe in bacteria, it's almost the opposite. Like what comes out and for testing is the losers of the battle.
Yes, exactly. So what you're seeing are the victims and the stretchers, the little bacteria carried out on their tiny little stretchers saying, well, this did not go well.
Right.
And that's what you're looking at. You're not looking at what's keeping quietly and hiding out in your crypts. The crypts are these nice pockets with mucus. They can dig in and they're hard to get out. So you know you've got villi in your small intestines that are like little fingers, and they are basically increasing the surface area of your gut so you can absorb more nutrients. But in between the villi are these little holes, these little pockets, and that's where the stem cells are. The stem cells live down in there. Your gut lining is being replaced every week, which is an astonishing amount of work. But there's a lot of stuff going through your gut, and a lot of damage is being done, and a lot of it's being done by bacteria. So you replenish your gut by having stem cells in these crypts move up the side, and the cells just keep being born in the basement and moving up to the higher floors every day. So this is happening every minute in your body. It's interesting when you look into the crypts, you find out that there's actually more oxygen in there, so that certain species of bacteria that like oxygen, the aerobes, actually find it nice and comfy deep in the crypt. You might think that that would be a place that would not have oxygen, but in fact, your body is producing it for these beneficial bacteria.
Is that gaseous oxygen?
Yes, it is actually in a form that can be absorbed by the bacteria. It's a little odd, isn't it?
Yeah.
It's not what you would think at all. There's one surprise after another as you start looking into this. And a lot of these studies are brand new. So this study that shows oxygen being generated in the crypts is maybe a year old.
So I've got to ask, is that gaseous oxygen typically fully oxidized? Because otherwise I would think that would lead to a lot of farting if you're actually creating gas that's not getting used.
No, it's not enough to do that. It's being absorbed almost immediately by the aerobic bacteria anyway. The gas that you're getting is actually from bacteria that is fermenting, and as it takes in the sugars, it produces these fatty acids and it produces gas. And the gas is many different bacteria produce different kinds of gas. So some of it's hydrogen, some of it's methane. All of those are perfectly sweet smelling and are not going to cause a problem. Other bacteria produce things with more sulfur in them, and those are stinky. But we probably need to get over our fear of farting, because
You heard it here first.
You heard it here. Because we have had refined foods for a long time, and refined foods produce less gas. But it's not clear that that's good for you. It's pretty clear that we're meant to be gassy and that we're meant to support a nice bacterial population.
Yeah, it's better to fart and not have type 2 diabetes than the other way around.
Bingo. Just choose your battle here, right? And go with the farting, make fun of it, do what you need. If you switch your diet over to beans, for instance, in Mexico they tell you what you say, well, how do I get rid of the farting? They say you just keep eating beans. It seems to get a little bit minimized once you raise your fiber levels to a normal level, but I'm not going to lie to anybody. There's farting involved.
We've been kind of spelunking in the gut for a while now. We talked about some mechanisms of how the gut is actually having its effects upstairs in the brain. Is there more we want to talk about there, missing puzzle pieces?
I think the only thing that's kind of interesting is to look at inflammation. And inflammation that is chronic in your body does affect your brain. And it's not totally clear why, but cytokines do get across the blood-brain barrier. And when they do, your microglia go into overdrive. And these are these cells that live in your brain, but they're kind of like amoeba. They are able to crawl around and scrounge up other bacteria that get into you past the blood-brain barrier. It's actually kind of crazy. The blood-brain barrier is poorly understood. We've always thought of it as like a fine sieve that keeps out all the big particles, and that's absolutely wrong. It's much more like an interface that looks at what's trying to get across and then says, okay, you can come through. You stand behind the rope. And so the bacteria, viruses, lots of things do get into the brain. They're not supposed to, and the microglia come after them and get them. But that's part of what is causing what we think is causing depression and anxiety and other mental issues. It's not just depression and anxiety, but you can lose cognitive effect when you have inflammation. The way that the hypothalamus works, there's something called the HPA, which is the hypothalamus pituitary adrenal axis.
Mm-hmm.
These three things work in concert. And when your hypothalamus, which is kind of like a tongue in your brain, it's one of the few places that is not behind the blood-brain barrier. It can actually taste your blood, and it can see when there's problems. And when it does, it talks to the pituitary, and the pituitary secretes hormones that reach the adrenal gland, and the adrenal gland starts up this fight or flight type thing. And the fight or flight response is very interesting. And I think this is behind a lot of our conversation today that we didn't really touch on, but the fight or flight is something that kicks in when you are, for instance, presented with a lion. So the lion says growl and you run. And you don't think too much about it, but what's happening is your HPA axis is actually shutting down your immune system. It says, I don't have time to fight that flu. Right now, I have to run from a lion.
Right.
And so this is actually one of the reasons why we have so much inflammation, is because we've kind of screwed up our HPA axis. And what turns out is that when you stop the inflammation, when you stop the immune response, you can actually let bacteria through. And the ultimate act is to let inflammation go up. So it's a tricky balance. And your body is always trying to find out how to get that done properly so that it can deal with your pointy-haired boss and or a lion and at the same time fight off the flu. But these are things that are kind of either or. And it's a fascinating thing to see that part of you shuts down when you are stressed. So stress is related to inflammation, which is also related to cognitive abilities and to depression and anxiety.
What studies are you watching now that you're most curious to see the outcome in the next, say, 6 to 24 months?
I'm looking at John Cryan and Ted Dinan out of Ireland because they are now moving on to human studies. So these guys are some of the most prolific scientists on the planet right now. I see a new paper coming from Cryan's lab about every week, and he has gotten a lot of money to do this research. He's got a lot of students that he works with, and they are just pumping out the research. So they're starting up their human studies. I can't wait to see what's coming out of it. There are preliminary results that I really don't think I want to talk about just yet, but there are some specific bacteria that they're finding.
Can you tell us anything about the study design? I know you can't talk about results yet, but just so we know what's being researched.
The design is pretty straightforward for these things. They're actually taking people that are depressed and they're measuring depression, and then they give them these probiotics and see if they are going to do better after the study is done. And some of the tests that they use are these written tests, which are standard ways that psychiatrists use to figure out whether you're anxious or depressed. But they're also using things like cortisol, where you can take a saliva reading and see what your stress levels are. When they put that before and after a probiotic test, they can start to see which ones are working and which ones aren't. It's pretty straightforward, but the numbers have to get higher. They have to get a lot more people enrolled, and you're starting to talk about some real money with human studies.
Right.
But it's fairly straightforward, and they're starting to get some stuff, and they are actually starting to nail down a couple of probiotics in particular. And the question is, will you be able to go to the store and buy these? And I don't think that's going to happen anytime soon. They just ran a study on 16 probiotic mixes that they found at the market, and I think 1 or 2 actually had in them what they said were in them. So you don't even know whether you're getting anything at all. You don't know whether you're getting live bacteria or whether you're getting the bacteria that are indicated on the jar. There are a couple of people who are producing. Johnson & Johnson actually seems to do something that is more believable. They have a probiotic that actually they've done randomized controlled trials on. We need more of those. And then you need to find out that these are actually truly what you think you're getting. So there's a way we have to go before all of this is answered. In the meantime, there are certain foods that you can eat that should make you happier. Among them is yogurt and other probiotics and synbiotics, which are prebiotics mixed in with probiotics. Things like sauerkraut is kind of more of a synbiotic than it is a pro or prebiotic. And then there are other foods that have lots of this fiber, and that includes artichokes, which we call fartichokes, and mushrooms and onions, leeks. These things are all high in something called fructooligosaccharides, and those are some of the oligosaccharides that feed the good bacteria. That's where we're at right now.
Smart Drug Smarts.
So thank you so very much to Scott Anderson for taking the time for that conversation about the finer points of our bacterial menagerie that we've got living in our guts. It sounds like an area where the science is moving extremely fast, and I think we can probably expect a lot of further developments, new findings, and potentially new therapies in the coming years. I was a little surprised to hear him say that so far there is not such a thing as poop fingerprinting, the way that they might scan a person's retina or their DNA or something like that to find exactly who this person is. If the investigators on CSI find a stool sample on the floor, they couldn't necessarily get the bad guy from that. It's also going to be hard to think of your poop the same now that you know that not only is your poop something like 10% dead bacterial husks, but those bacteria didn't die peacefully of old age, but were instead the losers in an epic battle taking place for the conquest of your colon. But anyway, moving right along to things that many of us have in common with schizophrenics, let's move to the ruthless listener retention gimmick.
Smart Drug Smarts, ruthless listener retention gimmick.
So one of the things that I find really fascinating about the brain and sort of our inner experience of consciousness is the insubstantiability of all of it. Basically, to find out what it's like to be somebody else, you need to ask them. We don't have a better way of really finding out at this point. I remember as a kid wondering if my version of the color yellow or any other color is the same as what other people see, because we could point at the same thing and say, yep, that's what we call yellow. But whether my yellowness is their yellowness, it just seems like an unbridgeable gulf. And there was an interesting study conducted just recently by a researcher researcher at New York University, looking at a sort of similar phenomenon of what it's like for people as they read. As you read something, do you hear an inner voice speaking the words or kind of reading it to you, or are you just drinking in the information without any sort of internal audio component to it? And similarly to another article that we spoke about recently where a researcher was getting data from looking at a bunch of YouTube videos, and that was sort of their data acquisition. In this case, the study's author, Ruvani Vilhauer, was looking at 136 representative answers to questions around this topic that she found on the website Yahoo Answers, which is apparently the world's largest English language Q&A site. And it turned out that there was maybe a surprising amount of variability in people's answers as to how they experience reading. A whopping majority of people, 82.5%, said that they did hear an inner voice when they were reading to themselves, whereas just over 10% said that they clearly did not, and the other answers were too ambiguous that she couldn't really categorize them clearly one way or the other. I myself definitely fall into the yes, I do hear a voice category. But there the demographics subdivide further. Some people hear multiple voices, like depending on the subject matter that they're reading, they might hear a man's voice versus a woman's voice, a stern voice versus a supportive voice, while other people hear only one voice, and very typically their own voice. It's just kind of like their inner speech track if they turn the volume all the way down. But of those people who heard voices, almost all of them described some sort of auditory component, like it had a particular tone or the volume might alter for emphasis at certain points. Some people did not like their voice, like they actually had a reading narrator that for some reason they found offensive or bothersome. That would suck. And interestingly, I should mention that this study was published in the journal Psychosis. Oftentimes when we think about people hearing voices, we think about schizophrenia, things like that. It's not necessarily something that people volunteer if they're hearing voices, because in most contexts it's considered pretty freakishly abnormal. Although there seem to be certain brain states where hearing voices is an absolutely normal part of the human experience. Really interesting book I'll drop called The Third Man Factor, if anybody wants to check that one out. It's about extreme survival situations, sort of people on the edge of almost dying in a horrible disaster, and kind of the bifurcation of consciousness that can kind of take place into sort of a mean boss and a servant that just carries out the boss's bidding. Something that happens so frequently in survival situations that it could not be a coincidence. But what's kind of fun about this study is that although it's something that could have been addressed at any point in history in a sufficiently literate culture where you could ask enough people this question, do you hear an inner voice while you read to yourself? It seems like nobody's really asked it before in a scientifically rigorous way. And the study's author speculates that it's because everybody sort of assumes that their own internal process, whatever it is that they experience, is kind of what everybody else gets too. That my yellow is your yellow is everybody's yellow, that kind of thing. But given that the inner reading voice is technically an audio hallucination that happens in 80+% of the population in psychiatrically middle of the bell-shaped curve type people, this could lead to some interesting avenues of study in the differences of what goes on in those inner voices when they become threatening, bothersome, or problematic in conditions like schizophrenia.
We like to think of ourselves as a digital speakeasy for brain hackers, but you can call us Smart Drug Smarts.
Okay, so you have made it to the very end of the episode, number 162. Thank you for hanging around with us. This episode and everything you might possibly want to know about it will be found online at smartdrugsmarts.com/162. If you missed last week's episode, we talked with Dr. Thomas Carmichael about how the brain repairs itself from stroke and how findings there might lead to future therapies. Next week, we're going to be bringing back a Smart Drug Smarts alumnus, Dr. Dominic D'Agostino. He's probably best described as a raging fan of human metabolism. Talked with him on a previous episode about ketogenic diets, and this time we're going to be talking about idebenone, which is also within his wide purview. That'll be next week, episode number 163. But as always, have a great week in the meantime. Hope the year 2017 is off to an excellent start. And as usual, stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.